everyone. So session 2 is around surveillance, detection and characterisation. And our keynote speaker is Joel Wertheim from the University of California. Thank you for coming all of this way and managing your jet lag and etc. So I'd like to introduce Joel, and his presentation will be on molecular epidemiology of the origin of SARS. Okay, thanks, Joel. Yeah, okay.
All right, is the mic working?
Yes, that's fine.
And then,
Thank you.
is this a clicker?
Yeah.
Excellent. So yes, I'm Joel Wertheim from the University of California, San Diego, and there we go. And this work represents a large international collaboration on 4 continents, exemplified by 2 papers that were recently published in Science. And I just want to, in particular, highlight this fine gentleman right here, who is my graduate student, Jonathan Pekar, who led the molecular epidemiology work. And of course, our funders, the NIH, CDC, and GISAID for holding on to all of this data for all of us, and funding from Google. Now, the origins of SARS-CoV-2 initially appear a little strange, admittedly, because the virus was first detected in humans here in Wuhan, in Hubei province, but the bats that are the natural hosts of the most closely related viruses are found in Yunnan province in China and in Laos, thousands of kilometers away, which seems a long way for a bat virus to travel, but on second blush, perhaps maybe not, because these viruses also are the natural reservoir of SARS-1, which appeared in Guangdong province and in Hong Kong, also quite far away from Yunnan. Also, it's worth pointing out that SARS-1 was also found in Hubei province in civets. So it's possible that animals can ride on trucks and move around the country. Now that we've dispensed with geographical confusion, let's actually talk about where we first saw this virus. And for here, we're going to go back to classic epidemiology. I've never presented this slide in England before. Anyway, so we can do the same type of thing with SARS-CoV-2, looking for the geographical locations of cases, particularly of SARS-CoV-2 in December in Wuhan, shortly after the virus was the first symptomatic cases were identified in mid-December. If we see, if we look at all of the WHO-defined cases, we see that they center very, very closely around the Huanan seafood market. In fact, the 1% contour of all cases found in the city in that month include that relatively small market in a city of 11 million people. What's remarkable is that if we look at all of the cases from December 2019 in Wuhan that had no epidemiological link to the market, here on your right, they also center around the market. So it's a pretty strong Broad Street pump-like signal. Now, what happens if we go, say, into the future, into January and February 2020, when the virus had firmly ensconced itself in Wuhan? Well, by then, there is no more geographical association with the market, and now it looks like this virus is just spreading throughout a large metropolitan area, and its geographical center has moved to essentially the geographical center of the city. So our signal for the market disappears as the virus spreads widely. Now, initially, it was unclear what role this market played because the WHO in their report said there were no live mammals being sold at the market, or wild mammals that could be susceptible hosts. But After that report came out, this paper, which we've already seen pieces of today in Scientific Reports, said, well, actually, we've been looking at tick-borne viruses in wild animal markets in Wuhan leading up to the pandemic. And yes, in fact, there were plenty of animals. Now, I'm going to take this slide one step further and just zoom in here because right there in figure D, that's a raccoon dog, an animal closely related to a fox, known to be susceptible to SARS-CoV or SARS-CoV-1, and we know it can, in laboratories, become infected and transmit SARS-CoV-2. And we have photographic documentation of it there in 2014 in the Huanan seafood market taken by Eddie Holmes, and we also see documentation of these animals in these markets through November of 2019, which makes me very suspicious because all of the animals were gone on January 1st, 2020 when the market was shut down. So we know that they were there in November of 2019. Then we start to see the first symptomatic cases of COVID. And then when the market is closed down, all the animals are gone. So yeah, we have no direct evidence of an intermediate host. None of these animals were ever seen nor sampled, but we know they existed and we know they disappeared. Okay. Also pointed out, this market is exceptionally well, not a smoking gun. We see a lot of red flags here because we see lots of positive environmental samples all throughout the market, specifically concentrated in the part of the market that was known to sell wild animals. So we have all of these indicators that this was a natural zoonosis from wild mammals being sold at the market. I can't tell you it's raccoon dogs because I don't know. There were lots of other animals being sold there that are potentially susceptible, including minks. But we see this unbelievably strong geographic association with a small corner of a market in a city of 11 million people for a virus that you would initially expect to be zoonotic. Okay, so that's where. What about the when? So when it comes to when, and if I want to know when, I start looking at the genome sequences. Now this is actually the SARS-CoV-2 genomic diversity through February of 2020, mid-February of 2020, A's, T's, G's, and C's, and as you can see, it is remarkably homogeneous. But that doesn't mean there's not signal. And to tease out this signal, we build a tree where we identify two major lineages of SARS-CoV-2, lineage A and lineage B, both of which are found in Wuhan. Now, a brief primer here. All you need to know about lineage B is that all of the human cases associated with the Huanan market were lineage B, and the earliest case was symptomatic on December 10th of 2019. As I mentioned, all of human cases there, and nearly all of the environmental samples at the market. Now, lineage B went on to dominate the epidemic, and all variants of concern descend from this lineage. There's also lineage A, its lesser-known cousin. Now, lineage A was not seen until late December, and it was seen near the Huanan market, but not at it. The first human cluster of SARS-CoV-2 in Wuhan was lineage A husband and wife and their son. Now, these individuals lived near the market, but they had not visited the market. Also, there was an individual, one of the first cases, whose genome was sequenced, stayed at a hotel near the market in late December, December 27th, but had no formal association or link with the market. Now, what's notable and surprising here is that although we saw lineage B first and lineage B spread more widely, lineage A is actually more closely related to, or more genetically similar to bat viruses. In fact, it is 2 mutations more similar. Now, 2 mutations may not seem a lot in other viruses, but SARS-CoV-2, where we can essentially count mutations and identify lineages by them, every mutation tells a story here. So we have a lineage A virus, which looks more like bats, but appears later. Why is that? And that's the question that we're going to answer today. And to do that, we're going to rely on our good friend here, the molecular clock. Now, for those of you who aren't evolutionary biologists, the molecular clock is just that steady drumbeat of mutations that accumulate over time, and in SARS-CoV-2, that drum beats pretty steadily. We can see here from next strain just that accumulation of mutations over time, about 2 per month, and using that, we can date evolutionary events within the history of SARS-CoV-2. So let's go and do that. We take our 800 or so high-quality genomes from the first 3 months of the pandemic, and we estimate the age of their ancestor. Now, not only can we estimate the age, but we can also, using this approach, say what was the genotype or variant of that common ancestor. Was it lineage B that dominated the pandemic, or was it lineage A, the one that was more similarly related to bats? Now, if we just look at this virus in humans, what we see is that lineage B actually appears to be the sort of genetic ancestor according to the molecular clock in humans. And matter of fact, we can strongly reject lineage A, the one that's more similar to bats, as that ancestral haplotype, which is surprising because naively you would think, well, it's more distantly related to bats. So the molecular clock doesn't like lineage A, which means that if you look in humans, we have to somehow resolve how do we get the bat virus later. So what I'd like to call your attention to on this early tree are 2 what we call large polytomies. A polytomy is a burst of transmission represented in the tree, where rather than 1 lineage leading to 2 and each of those lineages leading to 2 more lineages, we have hundreds of lineages all spouting from the exact same point on a phylogeny. And we see this at the base of lineage A and the base of lineage B. We've also seen this when the virus was introduced to new regions of the world that hadn't seen it before, like when the virus first got to New Orleans in the US or in Washington State, or when the virus B1 got to Lombardy in Italy. You see these massive bursts of transmission, massive diversification events before the virus has time to accumulate mutations. Now we want to know, how often would you see these, say, 2 massive bursts of transmission coming from a single introduction? So what we did is we simulated epidemics using this basically a complicated SIR model termed Sapphire, and then we simulated phylogenetic trees across those simulations and then went to infer those trees. And what we saw is that in 47.5% of our simulations, you see massive polytomies with 100 branches or more coming from a single point in the tree. Now this matches the base of lineage B and the base of lineage A, but it doesn't look like both of them combined on the same tree. And what about if we want to say, okay, let's have a massive polytomy at the base of the tree, and then we get 2 mutations and then another burst of transmission? How often do we see that in an epidemic simulation of, say, 5 million people on a scale-free network? Only 0.5% of the time. So it's very, very difficult for us to simulate, say, the introduction of lineage A, burst of transmission, the virus transmits slowly, and then another giant burst of transmission for lineage B. And that's the same if we assume that either A or B is ancestral, or if that ancestral viral genome was stuck in between them, one of these sort of intermediate unobserved genomes that gave rise to lineage A and lineage B separately. So we can't recapitulate the phylogeny of SARS-CoV-2 with a single introduction. We can recapitulate A and we can recapitulate B, both with these large bursts of transmission, but a single introduction can't account for both. Now, if we just impute if we sort of synthesize our simulations and our molecular clock inference and say, all right, then what's the probability that we could explain our data with 2 introductions, A separately from B, over the probability of just a single introduction given our simulations and the observed phylogeny, which you can sort of imagine like this. We have A and B separate over the probability of it being one of these 2 scenarios. And the answer is, well, we can actually strongly reject a single origin for SARS-CoV-2 based on these trees and simulations with a Bayes factor somewhere around 60, which not being a classically trained Bayesian statistician, I'm told this is pretty strong evidence. All right. Now I mentioned we can not only take the we can not only estimate sort of the shape of the phylogeny from the molecular clock, but also when it happened. Now our simulations allow us to push back the time ever so slightly from the TMRCA to say how long would it have been circulating in humans even before that TMRCA, to estimate not just the genetic age, but also when do we think these first infections would have occurred. And we can say that we think lineage B first got into humans around mid-November 2019, circulated for a couple of weeks before giving rise to its signal of its genetic ancestor. And then lineage A followed shortly thereafter, jumping into humans sometime in early December. Now, importantly, we see very, very limited evidence for cryptic spread before November. So even if the virus had jumped in, say, the very end of October, beginning of November, it couldn't have given rise to really tens or hundreds or thousands of cases before it was discovered. Because if that many people had been infected in Wuhan or anywhere in the world in October or early November 2019, we would see evidence of genomic diversity tracing back that far in time. And we just don't see it, which means, say, on November 15th or December 1st, there were only a handful of people infected with this virus. Now, that doesn't mean that we would have seen these first cases because, of course, not everyone gets sick with SARS-CoV-2, and not everyone would have been hospitalized, which is how we found these early cases. And so even though we only saw sort of that tip of the iceberg of sick individuals, there's very good evidence, based on limited genomic diversity, that we would have seen many SARS-CoV-1 or, lineage A or lineage B cases. Now, importantly, 2 introductions doesn't result in double the number of cases early in the pandemic, because as lineage B is circulating, if lineage A then jumps in, you've now only added 1 more case who can then go on to infect additional people. But it's not going to double the number of cases early in the pandemic. Okay. Now, why do we think that this is right? Well, shortly after we pre-printed this study, which predicted that both lineage A and lineage B were introduced separately, we got published or pre-printed evidence that lineage A was also at the market. And that came from the Chinese CDC, former director George Gao, who found lineage A, evidence of lineage A on discarded gloves right here in the part of the market where wild animals were being sold. Now, it's not necessary that this is evidence of animal infection, because this sample wasn't taken until January 1st when the markets were closed and the animals had disappeared. But it is evidence that someone in that part of the market was infected with this other lineage of the virus. And what's notable here, as I mentioned, is that lineage A is associated with the market, but not directly linked to it. And we don't see it until later in the month. And in fact, when we finally do start to see lineage A cases in late December, those 2 cases are closer to the market than the rest of the lineage B cases. So lineage B had already spread farther out into the city by late December, early January, than lineage A, which you can imagine like this. If you, say, have Huanan market as the center and you drop one zoonotic infection into it, a month later, lineage B has spread out this wide circle. But if you then drop lineage A in and measure it at the same time, it will be more tightly connected with the market, which is exactly what we see in the case data from Wuhan. All right. Now, I just said something kind of weird, because pandemics are rare. We get them about once a generation. And a pandemic of this magnitude that can essentially shut down the world and kill millions and millions of people, well, that happens once in a century. And I just told you that this once-in-a-century pandemic virus jumped into humans twice in 2 weeks. That's weird. I admit that at first blush, that seems counterintuitive. So, turn here to George Clooney, patron saint of zoonosis, and a guy who just seems to have all the answers. George, what do you got? Let's hope the sound works.
Right. Who are you looking for?
Tenzing Norgay.
Tenzing Norgay.
Tenzing Norgay. Who is that? Index case? Was he a seller at the Huanan market?
I doubt it. Tenzing Norgay was the Sherpa that helped Edmund Hillary climb Mount Everest.
How does that explain it? Well, we humans have existed in our modern form for hundreds of millions of years, and we've lived in the shadow of Mount Everest for tens of thousands of those years. And for tens of thousands of years, not a single human being had ever summited Mount Everest. And then, on May 29th, 1953, 2 men did. Sir Edmund Hillary and Tenzing Norgay. Not 1, but 2. Because everything that was needed in place to get 1 man up that mountain, the second also took advantage of that. Essentially, you have conditional probability. Getting the first person up is hard, probabilistically speaking. The second one is easy. So once you have a virus that is capable of transmitting in humans, and you put it in an intermediate host in close proximity to humans, the only barrier to a zoonotic virus with pandemic potential spreading is that final step, that final jump from an animal to humans, because everything else is taken care of. That rare virus has already evolved, and it has already established the fact that it can spread widely in humans and cause a pandemic. Now, the reason we didn't see it before, or the reason we stopped seeing these jumps on like SARS, the first SARS, is that they kept selling the animals for years, and we kept seeing multiple jumps into SARS. We keep seeing multiple jumps into MERS. SARS-CoV-2, those animals had disappeared by January 1st, and the zoonoses stopped. So, what do we think happened? I think there was a bat reservoir. We get a jump into an unknown intermediate host animal that was sold at the Huanan market. We think there was very shallow genetic diversity within this animal, much less than you see in MERS or in SARS-CoV. And then we think that there were 2 separate jumps. B gets in first, starts spreading and transmitting in humans first, which is why the molecular clock in humans wants to put the ancestor on B. Then that ancestral-looking virus, A, which hadn't picked up any mutations in the intermediate host, jumps in a couple weeks later. That's why the molecular clock in humans can't agree on the routing, and we can see this spread. Now, what's important here from a public health perspective is that if you have 2 separate introductions of this virus, it doesn't need to adapt and evolve in humans. As we saw before, when it infects mink and it infects deer, it infects cats, this is a generalist virus that's ready to go upon introduction. And we see evidence of that here with the 2 separate introductions of SARS-CoV-2. With that, I'll take any questions you might have. Thank you.
Thanks, Joel. Thank you. Any questions from the audience? Julian, I can see, and then Paul.
Hey, Joel.
Hi.
As always, lovely, lovely work. So when you go back and analyse the sequences from humans in the market, they're all lineage B, very, very, very small traces of lineage A, like 1 in 10,000 or something like that. How do you propose in the animal house that the lineage A was kept separate from lineage B? Presumably, lineage B, as you say, came from A, and that must have occurred in a transmission dynamic in a group of animals.
Yes. So I think what we have is a very small reservoir of animals at the market. And importantly, it doesn't even need to be one species, because as we see, if you stack these cages on top of each other, There's no reason to think that it could be that one species of animal introduced the virus to the market, but then you could have multiple different ones at the market infected. So you have multiple individuals, potentially of multiple species, and you have sort of this very, very shallow genetic reservoir. I don't think that it's all coming from a single infected animal. I think you're looking at sort of this very sort of small group of potential hosts, which unfortunately we've lost all the signal of, except for a series of positive environmental samples. Now, it's also worth pointing out that of the environmental samples that were tested by George Gao's group, they only got the distinguishing mutations between A and B in about 4 or 5 sequences. And of those 4 or 5, 1 is A. So by January 1st, those environmental samples, A isn't 1 in 10,000, it's more like 1 in 4, 1 in 5. So we really don't have this good idea. We know that earlier in the market, when you have a lot of human cases in mid-December, all we see is B.
Thank you. Paul?
What defines the geography of the human cases? With the epicenter centered over the market,
Yeah.
spreading out.
Okay.
Is that where they live?
Yes, that is residential address extracted from the WHO investigation, and that's in December. In January and February, that's people who are reporting COVID symptoms through a social media app. And we have their, or my colleagues have their residential information based on that.
So that implies people don't live very far from where they work in that city.
Well, of course, I mean, it is spread all, in January, February, it is spread all across the city. But yeah, we do see.
Get that tight focus over the market that you showed in your early slides.
Yes. Yeah, it implies that people are, I mean, that's not to say that you can't sort of work equidistant on both sides, but it's the center of that entire massive infection.
Dallin and then Andrew.
I was just interested in correlating your talk with what women have shown in mink farms in the Netherlands, and whether or not you considered the possibility that the virus had jumped from the intermediate reservoir into humans, back into the intermediate reservoir, and then back into humans in a way that might amplify the very first steps.
It is possible, although then you're sort of invoking how it why you would need, if it's in humans, why you would need to infect animals to infect more humans. And I think those are sort of unnecessary steps. I think what's most telling about the mink example and the hamster example is that once you have an animal infected with this virus in close proximity to humans, multiple jumps is what you should expect. We saw multiple jumps from hamster. We see multiple jumps from mink. We're seeing multiple jumps from deer. Once you put sort of that reservoir in close proximity to humans, this virus just likes to keep spreading. And then, of course, the question is sort of why of all places would you, if it was already in humans, would you see that geographical center of the market? And you sort of need to start invoking this, well, it died out everywhere else, and then it got there, instead of just look at SARS, look at MERS as sort of your reference.
Yeah, I guess the interesting thing with COVID, because it's got the broad host range, and it showed that in that very early step, the animal can in effect act as a human, in terms of the transmission potential. So you could have ping pong back between animal and human.
Yes. Yeah, and I think what's notable here is that all of the human diversity only dates back to when we would see sort of the association with market cases who were eventually hospitalized. So there's no evidence of, so if it were in humans, say, earlier, before we see these market cases, the genomic diversity would go much deeper into 2019. I think, and that's why they're just, it's not that scenario couldn't happen, and we see sort of it play out similarly in deer and mink, but there's no evidence of sort of other transmission chains of SARS-CoV-2. So either they all went extinct and happened to survive in this host and then get back into humans, or this is where it started. Now, that's not to say that it couldn't have jumped into humans before, say, upstream, and say from a rural area, because you would think people who were farming these animals could have been exposed, but of course, you put SARS-CoV-2 in a single human in a rural area, 95 to 99% of that time, it just goes extinct on its own because most people don't transmit the virus, especially if they don't have a lot of contacts like you would have in a city of 11 million people. So I wouldn't say that these is necessarily the first introductions of SARS-CoV-2 in humans, just the first ones that stuck.
Andrew?
Follows on slightly. I mean, why did the niche be then predominant? Any genetic factors?
Yeah. Okay, so I think that mutations between A and B are pretty meaningless in some debate. I think what happened is that, well, B got to Italy, and it also picked up D614G. Now, A eventually picked up D614G, but not for months and months later. And that sort of led to their first selective sweep in the virus. That's why, even though a head start had gotten a mutation like D614G would have come to predominate. D614G leads to B.1, which leads to all of the variants, and it got lucky and got a head start.
Okay, thanks, Joel. I'm sure there'll be plenty of other questions in the break. I'd like now to introduce Grace Tyson from University of Glasgow, who is going to continue our theme from earlier about talking about SARS-CoV-2 in cats. Microphone, please.
Thank you. Can you hear me okay? Cool. So, yeah, I'm Grace. I'm from the University of Glasgow. I'm a PhD student, and I am studying the serology of COVID in cats. So, a bit of background. There's many different feline species that are susceptible to SARS-CoV-2. It's not just domestic cats, but big cats as well. And as you might have been able to tell from Sarah's presentation, these has profound implications for feline health, but also for human health because of the danger of cats becoming a reservoir and the possibility of spillback into humans. We know that cats are able to mount an antibody response against both the nucleocapsid and the spike protein, and this means that serology can allow us to assess the prevalence of SARS-CoV-2 in the UK's feline population. So we use passive sampling, we use residual samples from the University of Glasgow's Veterinary Diagnostic Service, which had been submitted for a whole host of other reasons, detection of other infections, monitoring of illnesses, and pre-breeding and pre-export testing. And we used a pseudotype neutralisation assay with lentiviral pseudotypes to show how strong the neutralisation antibody response is. And this is superior to ELISA in a sense because it can actually show us the strength of antibodies rather than just their presence, as well as showing us the immunity to different variants. We used a panel of 4 different variants, the ancestral Wuhan strain, Alpha, Delta, and Omicron. So how this assay works, basically we incubate our serum with our pseudotype viruses. If the serum has no antibody presence, it should be able to infect our target cell, which are HEC293 cells expressing ACE2. The viruses have a luciferase gene in them, so luciferase will be produced upon infection, and when a substrate is added, we'll get bioluminescence. And conversely, if the serum does have antibody present, this will prevent the viruses from infecting the cells and will have no luminescence. So this means that we've got an inverted correlation between the amount of bioluminescence produced and our neutralising antibody concentration. And from that, we can calculate titers to tell us how strong the neutralising antibody response is in our samples. So our results, we've found that the seroprevalence of SARS-CoV-2 neutralising antibodies in UK cats was 3.2% overall. We tested over 2,000 samples, and this has increased over time. And in autumn and winter of 2021 to 2022, this was up at around 5%. These results aren't shown in the graph, but more recently we've had some results to show that this is slightly declining, and this might be due to waning immunity. We've also found that there's no correlation between sex and age in seroprevalence, but pedigree breeds do appear to be more susceptible than moggy cats. Interestingly, when we sorted the samples by dominance, so this basically means the variant which they produced the highest titer against. Although this we can't definitively prove without PCR data that this is the variant they were infected with, it does give us a bit of an idea. We found not only high levels of cross-neutralization of different variants, but also we found that the dominant strain in cats reflects the emergence of each variant of concern into the human population. And this could indicate that there's repeated cross-species jumps. The majority of cases do come from owners into their cats, so it appears that there have been repeated instances of infected owners infecting their cats. Interestingly, what we also found is we've detected extinct variants as being the immunodominant pseudotype in later samples. So you can see here in January of 2022, we're still detecting B1, the ancestral strain, dominant serum samples. So this could mean that there is quite long-lived immunity in cats, similar to humans. We also find distinct separate patterns of variant neutralization depending on dominant variant. So some conclusions from this. SARS-CoV-2 is well and truly circulating in the UK's feline population, and seroprevalence has increased over time and is at 3.2% overall. The detection of new variants mirrors the detection of the variant in the human population, but we are still detecting these dominant titers against extinct variants, leading us to believe that immunity is long-lived. There's high levels of cross-neutralization between variants. Infection with different variants elicits distinct separate patterns of neutralization, and because of this, we believe that both widespread serological as well as RT-PCR-based testing is vital for surveillance in cats and to address this One Health aspect of SARS-CoV-2 infection. And what would really help with this would be a wider case definition so that we can test more samples and have a more accurate population set, but also increased awareness not only among pet owners but also among vets so they can involve SARS-CoV-2 in potential diagnoses. So I want to say thank you so much for listening, and also thank you to everyone who's worked on this project and who's supported us throughout. I don't know if there's time for questions, but I'll take any if you have any.
One quick question. Louise?
Thank you. Thanks very much. Very interesting talk. Have you, or any class, or have you got any T-cell data? I'm just wondering, particularly on the memory cell.
So we don't have any T-cell data at the moment. We have obviously the neutralization data. We're also working on an ELISA to look for non-neutralizing antibody, which will tell us if there's just sort of weak antibody. This cat's still been exposed but doesn't have enough antibody. We don't have any T-cell data as of yet, but that would definitely be an interesting facet to explore.
Thank you. In the interest of time, we will move on to our third speaker for this session. And that's Ralfs Buks from the University of Aberdeen, who's talking about multiplexing and display platforms for immunodominant epitopes. Thank you, Ralfs.
All right. Should work, right? It's the other one. No. Oh, okay. I see. Thank you. Do you hear me well? Okay. I think I'll come here. So good morning, everyone. My name is Ralfs Buks, and I'm postdoc at the University of Aberdeen. And today I'll introduce you with our two technologies that will transform serology and antigen testing. Serology tests offer multiple advantages. They can be used to detect active and past infection. They can be used for epidemiological data, used for vaccine development, and immunity status checks. However, serology tests are not widely used in clinical setting due to modest sensitivity and specificity. And the limitations in serology tests stems from difficulty to identify B-cell immunodominant epitopes and the corresponding antibody class. There's no affordable technology that can multiplex different epitopes and antigens, and there is compromised specificity due to homology with other family members, and our two technologies addresses these challenges. The first technology is Epitope Predict. It is an AI system that identifies immunodominant epitopes and the corresponding antibody class. It is designed from experimental and publicly available data sets, and its advanced architecture means that the system is continuously learning meaning that the accuracy is continuously improving. But what we soon realized that we also need a complementary in vitro system that can multiplex, display, and correctly fold these immunodominant epitopes. And for this, we created our second technology, coined Epitogen, which is based when we bioengineered a naturally occurring protein. And we increased its stability and capacity to hold and display epitopes of interest. And the domain 1 and 3 naturally binds together, creating a stable stem-like structure that can display epitopes and nanobodies. It is non-reactive with human sera and other fluids. It can display epitopes and antibody fragments. It has unique binding properties, and it can display up to 100 epitopes, ranging in size from 5 to 700 amino acids. Because it is E. coli-based system, it has high efficiency, expression yield, and stability, and it's compatible with gold standard techniques such as ELISA and lateral flow. So we use these two technologies to develop accurate antibody tests for SARS-CoV-2, and our Epitogen technology offers 5 solutions to persistent problems in serology, which I will explain in the next slides. First, simply by putting antigen on our scaffold system improves the signal by 20% if you look at low signal samples. Epitogen is designed to bind preferentially to surfaces, meaning that the antigen or epitopes are exposed for increased signal. And it also binds preferentially to colloidal gold, which ensures correct folding for the lateral flow assays. And this will transform lateral flow technology because the major problem now is that antigens randomly bind to gold particles. And for the reason that Epitogen can also hold nanobody fragments, it can transform antigen testing as well. Second, we can use Epitogen scaffold to use it for epitope mapping. And when we look at overlapping peptide fragments for SARS-CoV-2 and test it using sera samples from COVID-19 patients, we can clearly observe some potential immunodominant epitopes. Next, when we check for antibody response to 5 immunodominant nucleocapsid epitopes, we can see a positive signal ranging from 50 to 80% of all samples. However, when we multiplex exactly the same epitopes on one scaffold system, we can detect close to 100%, and this just shows the importance of multiplexing to improve sensitivity of tests. We incorporated 16 mutations from spike protein, which increased the sensitivity by 1.5%. And last but not least, we included linear and conformational epitopes in our test. And just by doing that, increased the signal strength by 20%, also increasing the sensitivity. And as we thought that conformational epitopes are a big part of the overall signal to detect SARS-CoV-2. The 2 take-home messages is first, Epitope Predict and Epitogen are mature technologies in serology testing, and Epitogen will transform lateral flow and point-of-care test. And I'll just leave you with some recent press release articles about Epitogen, including a recent one from the United Nations. Thank you for your attention.
Questions for Ralfs before we break for coffee? If not,
or during the break.
Oh, yeah, or during the break. I've got a quick one. So in terms of the different types of epitopes, whether they be linear or conformational, have you looked at whether they're reactive versus neutralizing antibody characterization?
Oh, that's a good point. So we didn't look at it now. Our technology is based just for detection purposes now, but that's a really good point. Yeah.
Okay, thanks. Okay, we can break for coffee now, and please can people try to be back in the room by 5 past. I know we're running a little bit late, but I'll give you 20 minutes and then back starting at 5 past. Thanks very much.